So you should know the general gross anatomy of the digestive system, be able to label the organs of the digestive system in a diagram as you see here, or on a torso model. As well, again, no definitions and such, anything bolded within the exercise 25. What you see here goes along with figure 25.1 on page 312. Again, no periodal versus visceral peritoneum, and know where you find omentum and mesentery proper. Again, omentum, you've got the lesser and greater omentum that are attached to either side of the stomach, the greater and lesser curvatures, and you have mesentery that extends out to the small intestines. Okay, so if you remember, omentum is mostly fat. It is an insulating fat drape, as you see there. It just covers the anterior side of the abdominal cavity. Mesentery delivers blood vessels, nerves, and lymphatic vessels to the small intestines. Very little fat in mesentery. So know the difference between the two. And again, a mesentery as a general term is a dual-layered serous membrane, as it comes off either the visceral peritoneum or the periodal peritoneum. Mesentery proper is that specific mesentery that delivers vessels and nerves directly to the small intestines. So this slide, you want to know the 4 layers of the alimentary canal. As you start from the lumen out, you have the mucosa, submucosa, muscularis, and serosa. You should know characteristics of each of those layers, as you see on table 25.1, page 313 in your lab manual. Be able to identify those layers based on either a description or a diagram like you see on page 313 or here. Mucosa, again, you should, as part of the description, know some of the sub layers, but with mucosa you have an epithelia and a very thin muscularis layer. Often you're looking at the ducts of glands and things like that within the mucosa layer. Submucosa layer, you have actually the gland itself, blood vessels, and there is a thin nerve plexus between the submucosal layer and the muscularis layer. As you see there, the enteric plexus is divided into the myenteric and submucosal plexi. Muscularis layer, again, know that you have 2 layers, a circular layer that reduces the diameter of the lumen when it contracts, and a longitudinal layer which causes the tube to shorten up, but the lumen get larger. The serosa is also known as the visceral peritoneum. Again, a concept you see with the heart as well. The visceral peritoneum is also, or the visceral pericardium, the visceral pericardium is also the epicardium layer when you're looking at 3 layers of the heart. Likewise, the outer layer, if you're looking at the tube as the alimentary canal, is also the visceral peritoneum, the layer that covers the organs when you look at the visceral versus the periodal peritoneum. Know the general anatomy of the mouth, the oral cavity, hard palate versus soft palate. The fauces, again, is the opening. The posterior of the oral cavity is the opening into the oropharynx. And again, just know general teeth, incisors, canines, premolars, molars, things of that nature. And I did mention the frenula, the upper and lower labial frenulum, and then the lingual frenulum holding the tongue. Just kind of know those structures and what you see here. So with the teeth, again, no need to know specifics. Again, just know the types of teeth. Just know that as you progress from medial to lateral, it goes from incisor to canine to premolars to molars. I won't ask you specifics about numbers or things like that. You should know the general anatomy of a tooth. You also see this on figure 25.9 in page 319 in your lab manual as well. They just have a generic crown, whereas this diagram has a clinical and anatomical crown. But the crown is that part of the tooth that projects above the gingiva. If the gingiva start to deteriorate, the clinical and anatomical crown become the same in this diagram. Just know, be able to label what you see here. And again, just know things like enamel versus dentin, the periodontal ligaments, and again, the crown versus neck versus root, things like that. So with the anatomy of the stomach, again, just know the things that you see here: the esophagus, the gastroesophageal opening versus the pyloric sphincter. The fundus, again, is that large part of the stomach that turns back and is superior to the gastroesophageal opening. It kind of turns into the body, which narrows into the pylorus, or what you see is the pyloric part here. Again, another important thing to know about the stomach, it has the same 4 layers that you see in the rest of the alimentary canal: the mucosa, submucosa, muscularis, and serosa. But here you have a third muscularis layer. You have the oblique layer, as well as the circular and longitudinal layers. So again, just know the general anatomy of the stomach. The stomach continues on, and you should know the parts of the small intestine. The duodenum or duodenum, tomato, is the first foot or so. The jejunum is the next, I believe 8 feet or so, and the ileum is approximately 12 feet in length. The ileum connects to the large intestines, specifically the cecum of the large intestine, at the ileocecal junction. And then you see the mesentery proper as well. Again, mesentery is mostly a conduit for vascularity. Omentum is a storage area for fat, adipose. So you should know 3 mechanisms that the small intestine uses to increase surface area. Again, if we are absorbing across the membrane, the greater the surface area of the membrane, the greater the amount of diffusion can occur across that membrane, or absorption. So the 3 things that we are looking at to increase surface area, the one you see on this slide, actually the 2, you see the circular folds are just longitudinal folds within the small intestine, kind of if you were to take a stretched out tube sock and to bunch it up. It's still, the same surface area, but it's much shorter because of the folds. The other thing you see, microscopically, you see villi, little finger-like projections that you see at the bottom of the slide here. And you see villi at the top of the slide here. The villi, again, are little just back and forth individual fingers, so they dramatically increase surface area. You have 2 important structures within the villus. You have the lacteal, which is a dead-end tube from the lymphatic system that absorbs fats and lipids. And you have a blood capillary. You have an artery and a vein, basically. The artery drops off oxygen, and the vein is loaded with carbon dioxide that has been picked up from those cells. The other thing that is important to know about this venous blood that comes out of the small intestines, it is loaded with nutrients. So all these nutrients that you have just absorbed across your small intestine, you will find in the venous blood that leaves the mesentery. The mesenteric vein is loaded with nutrients. And the third thing we see, the villi, again, the cells line the villi, but if you look at the individual cells themselves and magnify the cell membrane, the plasma membrane of each cell has these finger-like projections that go back and forth that, again, dramatically increase surface area. The plasma membrane invaginations are just known as microvilli. So villi, yes, they are microscopic, but they're a much larger scale than microvilli. All right, microvilli are invaginations of the cell membrane. Villi are the little finger-like projections that you can see within the small intestines. So know those 3, the circular folds, villus, and microvillus, and know the difference between the 2. or sorry. Know the structure of the liver. Left lobe and right lobe are fairly simple in the falciform ligament on this anterior view. In this inferior view of the liver, you have the other two lobes, the quadrate and the caudate. Just know the right and the left lobe. But what's important in this view is the porta, the hepatic porta. There are three structures in the porta that are replicated microscopically at the portal triad at the corner of each lobule within the liver. You have a hepatic duct, which is draining bile. Bile flows out of the liver, and you have two inputs of blood. You have the hepatic artery, which delivers arterial oxygenated blood, and you have the hepatic portal vein, which delivers deoxygenated blood fresh from the small intestines, which, if you remember from the slide before, as blood leaves the small intestines, it may be deoxygenated, but it is loaded with nutrients. So that nutrient-rich blood from the small intestines first goes to the liver for conversion, detoxification, things like that, before being delivered to the rest of the body. So know the structures that you see here, especially as they relate to the porta of the liver. So if you look at the bile ducts and the hepatic ducts as they leave the liver, the common structure here you see you have a right and left hepatic duct. As soon as they merge, the short length is known as the common hepatic duct. Again, anytime there's a branch point, there will be a name change. The duct draining the gallbladder is known as the cystic duct. The cystos refers to a bag, and where the cystic duct meets the common hepatic duct, from that point on, they're known as the common bile duct. The common bile duct merges with the pancreatic duct at the hepatopancreatic ampulla and pokes through the duodenum at the major duodenal papilla. All right, so this allows for both bile and pancreatic juice to be ejected into the duodenum during digestion. So have a feel for the anatomy of the bile ducts as they go from the liver to the duodenum. So microscopic structure of the liver looks like what you see here. You have these six-sided hexagonal columns known as liver lobules. At each corner, each of the six corners at the edge of the lobule, you have what's known as a portal triad. The portal triad, just like the porta at the gross level of the liver, contains a hepatic duct draining bile outwards, and a hepatic portal vein and a hepatic artery, both bringing blood into the lobule. So the oxygenated blood from the aorta and the nutrient-rich blood from the portal vein merge and coalesce as they travel from the portal triads to the central vein. They merge through what's known as a hepatic sinusoid. The sinusoid is just an open area where two blood supplies are allowed to kind of merge. It's kind of a large open blood supply area. The hepatocyte are the cells, and the hepatocytes are arranged in what's referred to as hepatic cords, like little spokes of cells as they radiate outward from the central vein. The other thing you see there again is the bile canaliculi. So the hepatocytes are producing bile that flows in the opposite direction of the blood, and they're also digesting nutrients in the blood and producing chemicals in response to toxins and things of that nature. So again, know the microscopic structure of the liver, and definitely know the three structures that fall within the portal triad. All right, that is an important kind of concept to understand here. So again, refreshing blood flow through the liver. So as blood comes from the aorta, you have blood that can go directly into the liver at the hepatic artery, delivering fresh oxygenated blood. So as you see at the bottom at the porta of the liver, you have the hepatic artery, the hepatic portal vein, which has just brought deoxygenated but nutrient-rich blood from the small intestines, and you have the hepatic ducts, which are draining bile out and out of the liver into the intestines to aid in digestion of lipids. So again, as you see here, between the porta and the portal triad, and within the lobule itself, blood and bile flow in opposite directions. That is another important concept you want to know. Blood and bile flow in opposing directions. So the porta of the liver, again, you have the three structures that you see replicated microscopically at the portal triad. Each portal triad, you have a hepatic artery branch, a portal vein branch, and a hepatic duct branch as well. So through the sinusoids, the portal vein and hepatic artery vein, portal vein and hepatic artery blood coalesce and come together through the hepatic sinusoids. Again, the sinusoids dump into the central vein. All the central veins eventually coalesce into a hepatic vein, and the hepatic veins dump almost directly into the inferior vena cava. If you look at the anatomy below, the liver almost totally encapsulates the inferior vena cava at a certain level. So just again, no blood flow through the kidney, and know the significance of the portal triads. So just know your basic pancreas anatomy. Again, I would focus on knowing the bile duct, hepatopancreatic ampulla, pancreatic duct, things of that nature, and know what part of the small intestine the hepatopancreatic ampulla dumps into. All right, that's the duodenum that you're looking at, that first little foot that curves back into C shape from the stomach. So you should know the structure of the large intestines. Again, I would focus on knowing things such as the cecum, the appendix, ascending colon, hepatic or right flexure, transverse colon, splenic or left flexure, descending colon, sigmoid colon, rectum, and anal canal. Okay, you should know those structures and the order in which they arise as you travel through the large intestines. So this slide goes along with page 321, figure 25.10 in your lab manual. You should know basically the enzymes that digest the different groups of nutrients. Alright, so if I gave you enzymes that were named amylase disaccharidases, or as your lab manual mentions, what's called brush border enzymes within the small intestines, you should know that those digest carbohydrates. If I gave you trypsin, chymotrypsin, pepsin, peptidases, you should know that those digest proteins. If I gave you the name lipases, bile salts, whether that lipase is from the lingual area or the pancreas, pancreatic lipase, just know that those digest lipids. Okay, fairly straightforward, but just know the enzymes that digest the particular groups of nutrients.